Fantastic. Well, hi everyone. I'm really happy to have been given the chance to speak to you today at the January Colour Group meeting. And as Andy said, the title of today's talk is a little bit of a change from what was in the original programme, and it's going to be on the implementation and evaluation of multi-primary high dynamic range displays for vision science. And I hope this is still going to be of interest to everyone. So an outline for my talk today, I'm going to start by zooming out to the big picture to motivate multiple primary displays in general. And to do that, I'm going to discuss what it is that we're actually trying to reproduce when we're thinking about reproducing scenes on a display, and why we need a display with more than 3 primaries in order to do this. I'll then introduce the RealVision MPHDR display, and finally discuss ways in which we can quantify the capabilities of multi-primary displays, such as the RealVision MPHDR. So let's dive in. So what is it that we actually are trying to achieve with displays? Well, ultimately, we're trying to reproduce the light from a real world scene, but more specifically, we're trying to replicate how the light is sensed by the human observer. So we need to consider how the observer senses light. And as you all know, for a human observer, light is sampled by the 5 photoreceptors in the retina. This is the 3 cone types, the rods, and melanopsin, which is found in intrinsically photosensitive retinal ganglion cells. And again, as you all know, each of these 5 photoreceptors has a certain spectral sensitivity. That's the probability that they'll absorb light of a given wavelength. So that when a light spectrum is sampled by these 5 photoreceptors, it's reduced to 5 photoreceptor signals. And this is all that's available to downstream neural mechanisms. So really, it's these 5 signals that we care about reproducing on display, as opposed to the full light spectrum that elicited these signals, as these signals are all the observer has access to downstream. And broadly speaking, the 5 photoreceptors have different functions, with the cones and rods primarily driving visual processing, including form, color, motion, depth, and visually mediated behavior, with the cones primarily responsible for photopic processing and the rods for scotopic processing, while the melanopsin-containing IPRDCs drive non-visual light-mediated mechanisms. This is things like melatonin suppression in the evenings, which helps drive sleep-wake control, the light-induced entrainment of circadian rhythms, the pupillary light reflex, as well as alertness and mood. And there's growing literature on the function of melanopsin, as well as the cone and rod inputs to IPRGCs, and on the possible influence of melanopsin on vision. I don't have time to cover all of this in depth today, but the point I really want to make here is that there's many more neuromechanisms driven by light than just traditional photopic vision. And these functions are driven by the inputs of these 5 independent photoreceptors. And importantly, these inputs are overlapping. So traditionally, these functions associated with rods, cones, and IPRGCs may have been believed to have been more separate. So they may have been studied by different groups of researchers. So a color vision expert might not have studied IPRGC function, but it's becoming increasingly apparent that the photoreceptors do interact. So the only way to study them holistically is via a system that can independently control all 5 photoreceptors. And basic linear algebra tells us that if we want to do this, we want to independently control the activations of the 5 photoreceptors, we need control of 5 independent channels on the display. But the vast majority of commercial displays, if not all commercial displays, use only 3 primaries. So it's impossible for these 3 primary commercial displays to control these 5 photoreceptors. So by design, they're designed to control just the cone responses and don't offer any control of rod or melanopsin function, which means that while many displays have excellent visual reproduction and in many cases can reproduce the colors and shapes of a real world scene very well, the non-visual light response is ignored and uncontrolled. And this basically means that while we might be able to reproduce a scene that looks indistinguishable from a virtual environment, the real world scene compared to the virtual scene will have different non-visual light mediated mechanisms. And what's more is that there are no existing standard metrics that describe the reproduction or lack thereof of these non-visual features. So things like color gamuts and resolution only describe the reproduction of the cone signals. This leaves 2 open questions for future display technologies. The first being whether we can control the non-visual light mediated neural mechanisms with displays. And in order to do this, we first need a better understanding of melanopsin function in humans, particularly how the melanopsin signal is integrated spatially and temporally. And to this end, we've developed a research-grade system in collaboration with our RealVision ITN colleagues from Rafael Mamtiuk's group at the University of Cambridge, which we call the RealVision Multi-Primary High Dynamic Range Display, or MPHDR for short. And the MPHDR offers control of the 5 photoreceptors in isolation, spatiotemporally, and over high dynamic range. And the second pressing question for future multi-primary display design is what metrics can we use to appropriately evaluate these displays that consider the reproduction of rods and melanopsin signals as well as reproduction of the cone signals? And we propose a novel metric as a sort of extension of a color gamut into the 5D photoreceptor space, which we call the photoreceptor signal reproduction metric. So first, I'll speed through the capabilities of the MPHDR display. And if you're interested in reading about it in more detail, then I'd direct you to our recent Dataset publication, which describes the design and verification of this display. So the RealVision MPHDR is the only display of its kind. It allows for high dynamic range control, spatiotemporal control, and control over multiple primaries, which existing commercial displays and HDR displays don't. They only offer 3-primary control. And most systems which are designed for melanopsin isolating experiments, 5-primary tools that are custom built are Maxwell view systems which lack spatiotemporal control that the RealVision MPHDR display offers. So really with this system, we're going to be able to study melanopsin in isolation in ways that have been previously unachievable just due to the limits of existing display technologies. So how does it work? Well, the basic design of the MPHDR is to set 2 HDR displays on top of each other. So instead of a single projector backlighting an LCD screen as in a normal HDR, we use 2 projectors to backlight the LCD screen. And the color wheels in each of these projectors have been removed. So they each just have a single spectral output. And we place notch filters with the following spectral compositions in front of each of these projector wheels. And the choice of these filters was based on optimization of the resulting color gamut and melanopsin contrast from the overall display. The light from these 2 projectors is then used as a replacement of the backlighting of an iPad LCD screen, and diffusers and Fresnel lenses are used to distribute the backlight over the LCD. The observer then views the display from this sweet spot position, where they get the maximal overlap from the light output from each projector, and this is the position from which the display gets calibrated. So here is the display in all its glory. You can see here the 2 projectors with the filters in front, and here it is fully enclosed to prevent stray light escaping. So to present a stimulus on the MPHDR, the experimenter has control over 5 parameters depicted here in this vector kappa, which are the intensities of the 2 projectors, KE1 and KE2. And remember, as the projectors have had their color wheels removed, the experimenter can only vary the monochromatic intensity of each projector. And because of these filters, the resulting impact of this on the spectral output will be different from each projector. The other 3 parameters that the experimenter can control are just the intensities of each of the 3 primary channels of each LCD pixel. So for each pixel, like with the normal display, they can set the red, green, and blue intensities on the LCD. This means that at any spatial location on the display, the resulting spectral output is simply the product of the projector emission spectrum and the LCD primary transmission spectrum, weighted by these experimenter-defined intensities. But as it's quite tricky due to the spatial configuration of the display to measure the projector emission spectrum and LCD transmission spectrum directly at each spatial location, we can instead measure the product of the projector spectra and the LCD transmission spectra from where the observer would view the display, and we call this an effective primary of the display. And we can substitute this effective primary back into this equation for the spectral output of the display to get an equation for the output spectra of the display as a function of the effective primaries and these 5 independent parameters that the experimenter can control, i.e. this is expressed as a function of quantities that can be easily measured or specified. And just to show you here the spectra of the resulting effective primaries we end up measuring, and the full equation for the output of the display. This gives us the following color gamut from the MPHDR display, but due to the interdependent control of the primaries, it's not possible to cover every single region bounded by this convex hull of the gamuts. And here I'm just showing photographs of the display when only the top projector is switched on, the bottom projector, and both are switched on, just so you can see the differences caused by the different spectral outputs of the 2 projectors. And here's the 3D color gamut of the MPHDR with both a log and linear luminance axis in CIE XYY space. And you can see the peak luminance output of the display at the sweet spot is some 3,200 candela per meter squared. So the other key element that makes the MPHDR unique from other systems is its high dynamic range. So here I'm just showing the global and ANSI contrast of each of the components of the display, as well as the 2 HDR subsystems within the display and the full MPHDR display itself, which has a resulting dynamic range of over 3 million to 1. So what sorts of experiments can we run on the MPHDR? Well, it's designed in the first instance to run melanopsin isolating experiments. So we can use an optimization approach combined with our driving algorithm, which I just described, to find pairs of spectra, which we call metamers. So here I'm just showing, as a reminder, the spectral sensitivities of the 5 photoreceptors and such a pair of metamers. And these metamers give equal activations in the 3 cone signals as each other. So we can see this here in the bar graph, but differing rod and melanopsin responses. So here I'm just showing the contrast, that is the percentage change in activation between the photoreceptor signals from the two spectrum. And you can see in this example that the cone signals are matched between the two, while the rod and melanopsin signals are different. And we can make use of this experimentally by fixing the cone signals and varying the melanopic content of the scene on the MPHDR to assess the role melanopsin is playing in a particular mechanism that we might then subsequently measure. And here I'm just showing proof of principle of the display's capability to perform these melanopsin isolating experiments. So I'm showing an example of a pair of such metamers that were chosen by our optimization approach to be shown on the display at 1000 candelas per meter squared, with one spectrum containing a high melanopic content and the other a low melanopic content. And the spectra that we want the display to show are shown in the dashed lines, while the spectral output we actually measure are shown in the solid lines. And we can see the two clearly overlap, so the display is able to produce this pair of metamers that we generate. And we can see in the graph here on the right, where I'm plotting the Melanopsin contrast on the y-axis and against luminance on the x-axis over a luminance range of 75 to 2000 candelas per meter squared, that the requested contrast in black and measured contrast in red strongly agree across this relatively large luminance range. And here I've picked the metamers that can provide the maximum theoretical contrast at each luminance level. So we can see at low luminance, the display reaches a melanopsin contrast of around 120%, and at high luminance, a contrast of still around 25%, which reflects the fact that the gamut was narrowing at these higher luminances, but means we're still able to achieve a relatively high contrast at these high luminance levels. So the next steps here would be to demonstrate this proof of principle psychophysically and to run these melanopsin isolating experiments that the display has been designed to do. But unfortunately, the pandemic has gotten in the way of that a little bit. So the first set of experiments we plan to run now that we're back up and running in the lab is to measure the pupillary light reflex as a function of luminance at high and low melanopic excitations, so that we can really unpack the impacts of melanopsin and luminance on the pupil response. In terms of development, one of the major next routes we'd like to take is to move from the current 6 effective primary, 5 independent parameter model to a fully 6 independent primary driving model for the display. So this might involve the use of, say, temporal dithering to achieve primary independence at the expense of some temporal control, or a redesign which uses a monochromatic LCD with color DLPs rather than the other way around as we have it in our current system. Okay, so I want to switch gears a little bit now and talk about metrics for evaluating such multiple primary displays. So for the MPHDR display, for example, the color gamut doesn't fully capture all of the capabilities of display. You need to also look at the melanopic contrast available to understand the full capabilities of that display. Which begs the question, well, are there any metrics that might better describe the full capabilities of multiple primary displays beyond color gamuts? Or how can we extend color gamuts into 5-dimensional photoreceptor space? And such metrics are going to be really important for informing future display design, and particularly primary choice. So which primaries do you choose to maximize performance of your multiple primary displays? But there's no obvious equivalent to a color gamut or the CIE-XY horseshoe diagram in 5-dimensional photoreceptor space. So our approach, which is discussed in more detail in our recent bioRxiv preprint, is to propose a move to a photoreceptor-based chromaticity diagram and photoreceptor-based metrics, which you'll all be very familiar with, as photoreceptor-based diagrams and metrics offer a more natural extension into the melanopic axis. And this also follows the broader move within the CIE towards a cone-based model of colorimetry. So we take the familiar McLeod-Boynton equiluminant chromaticity diagram as our basis, which plots the S over L percent against the L over L percent signal. And we can add an additional axis to this diagram, which is the melanopsin signal over luminance, plotted against the L over L percent signal in the middle and against the S over L percent signal in the projection on the right. And here I'm just showing the spectrum locus in each of these projections along with the daylight locus in each of these projections. So what we end up with here is 3 2D projections of this new kind of 3-dimensional space that we've defined. The other challenge in extending into 5-dimensional gamuts is deciding what reference data set to use. So for chromaticity, for reproduction, for example, we don't quantify how much of the spectrum locus is covered by our display directly, because we're still quite a way off achieving this. So what we do more often is compare our display to reference gamuts like sRGB and Adobe RGB, or to reference data sets like reflectance samples from the color rendering index, for example, which are used as standard references. But As there's no obvious equivalent reference gamut in 5-dimensional photoreceptor signal space, we take the reference dataset approach using the definition of the color rendering metric as the basis of our quantifications. So we use the 99 reflectance samples from the ITU standard, as these are the ones used in the most recent definition of the color rendering index. So they're already accepted as a standard for display reproduction. And we rendered these samples under the 401 illuminance from Hauser et al., as these are the real illuminance under which these samples are rendered in the definition of the color rendering index. And we use every combination of these for our radiant spectra, which I'm just showing the chromaticities of here in our CIE XY horseshoe diagram. And here I'm just showing these spectra in the McLeod-Boynton diagram, which you'll all be familiar with, and our extensions of the McLeod-Boynton diagram, where the heat map here shows how many spectra fall within a given region of chromaticity space. Now, it's important to be upfront about the limitations of the dataset. So this dataset only contains matte surface reflectances under an illuminant. So what this dataset doesn't contain are things like structural colors and interference colors. And when we think about trying to reproduce real world light, which is how I started off motivating, what we're trying to do on display is reproduce real world light. Well, the vast majority of real world spectra are likely to be matte surfaces under an illuminant, things like leaves, skin, buildings, etc. But structural colors like the feathers on a peacock, while less frequent, are particularly salient in our perceptual experience, and they're not considered in this data set. Really, what we'd ultimately want is a better characterization of the spectral diet of the human observer if we want to improve our reference data set. So what sorts of spectra are people actually interacting with? But we take this as a good starting point as the sort of spectra you want to reproduce on a display. And we take some example displays to quantify with our proposed metric. And here I'm just showing the primaries of those displays. So we have 3 real 3-primary displays, and these are the measured spectra from a CRT and Dell LCD monitor and the CRS display++. And we take 2 5-primary displays, which are hypothetical displays, 1 narrowband, 1 broadband. We take theoretical 5-primary displays as there's no commercially available 5-primary displays to use as a standard. So we can first quantify these displays with our real-world reference data set by looking at just traditional chromaticity reproduction, this being the number of real-world spectra that can be reproduced within the chromaticity gamut, divided by the total number of real-world spectra in our reference data set. And we can see that all of the displays, both 3 and 5-primary, have high chromaticity reproduction, which I'm showing here in this bar chart, where the 3 3-primary displays are shown in purple and the 2 hypothetical 5-primary displays in pink. And they all have high chromaticity reproduction. And we can extend this then to the 5 photoreceptor signal space. So instead of quantifying the percentage of real world colors that can be reproduced on our display, we can quantify the percentage of the 5 photoreceptor signal quintuplets that are generated by the real world spectrum that can be reproduced on the display. And we call this the PSRM, which stands for the photoreceptor signal reproduction metric. So this is how many of the 5 photoreceptor quintuplets can be reproduced. And unsurprisingly, we find with the 3 primary displays, which are these purple bars here, which we can barely make out, that they have very low PSRM. And this is to be expected, right? Because any reproduction of the full 5 photoreceptor signals on 3 primary displays will just be by chance alone. They're not controlling the reproduction of the rod and Melanopsin signals. While for the 5 primary displays shown in pink again here, they have a high PSRM, which is what we'd expect. The other thing I want to point out here is that the PSRM is different for each of the 5 primary displays. So the virtue of having 5 primaries alone isn't enough to reproduce all of the real world photoreceptor quintuplets. What those primaries are is important too. I should further note here, for these purposes, we're only interested in the relative photoreceptor quintuplets, as the vast majority of displays can't reach the dynamic range of the real world. So this is just about reproducing the relative signals, not about reproducing them at an absolute intensity. Also, a note of caution for thinking about the PSRM metric is that with any metric, the usefulness of it depends on the intended purpose of the display and what you are using that metric for. So if we apply the chromaticity and photoreceptor signal reproduction metrics to the MPHDR display I introduced to you earlier, we see high chromaticity reproduction, which we'd expect because we optimize the choice of the design of that display to maximize chromaticity reproduction. But we have a relatively low PSRM for a 5 primary display. And this is because this system wasn't designed to maximize the PSRM, but rather to offer maximal Melanopsin contrast over a large luminance range for the research purposes intended for that system. And this is important when we consider commercial applications too, because we might not always want to maximize the PSRM. We may have other things that we want to maximize in our display. So it's always important to consider what the goal of your display is and how appropriate the reference is for that. So here we can see the photoreceptor excitation diagrams again, depicting visually the PSRM of the displays. So here in the bottom panel, I'm just showing all of the spectra in our reference data set again on the extended McLeod-Boynton space. So the familiar McLeod-Boynton axis here, and then the melanopic against L over L plus M, and the melanopic against the S over L plus M here. And in the top panel, I'm just plotting the spectra which are reproduced on the display plus, and I'm just taking the display plus as an example of a 3-primary display. And you can see the patchy reproduction of the 3-primary displays, which is to be expected as there's no explicit control of the rod or melanopsin signals. So any spectra that are plotting here are just plotting by chance. They just happen to reproduce the rod or melanopsin signals when you control the cone reproduction. Whereas in the middle, I'm plotting the spectra that were reproduced on the narrowband 5-primary display as an example of just a 5-primary system. And we can see we have good coverage of the real world spectra in our diagram. The other nuance to point out is that these 3-primary displays don't just fail to reproduce the rod and melanopsin signals from the spectrum, but they produce a distorted rod and melanopsin signal. So to illustrate this, if we imagine trying to show this spectrum from the real world on this theoretical 3-primary display, well, to get the cone signals to match, we'd need to give this spectral output to this 3-primary system. And if we then plot in the darker bars on the left, the photoreceptor signals from this real spectrum, and in the lighter bars on the right, the photoreceptor signals that we get from the display output spectra that is required to get the cone signals to match, that while the cone signals match between the real spectrum and the display spectrum, the rod and melanopsin signals are relatively lower in the displayed spectrum than the real world. And we can quantify this distortion with an additional metric, which we call the PSDN for the photoreceptor signal distortion metric. And this quantifies the contrast between the reproduced signal and the real world signal that we were trying to reproduce. And we can calculate this across all of the spectra in our reference data set and plot this as, say, a box plot to show the average distortion for the rod and melanopsin signals and also the spread of this distortion. And we can also visualize how this distortion changes with chromaticity by plotting the mean distortion for all spectra that fall in a certain region of the CIE XY chromaticity diagram, which I'm showing here for rods on the left and for melanopsin here on the right. And this black dot in the middle just represents the chromaticity of D65. And the main takeaway here is that with the 3-primary displays, we introduce quite large distortions in the rod and melanopsin signals, which varies as a function of chromaticity. So it's not just that we're reproducing the rod and it's not just that we fail to reproduce the rod and melanopsin content of the real world scene, but we're reproducing some screwy rod and melanopsin signals, i.e. the rod and melanopsin signals that would not arise in combination with that cone triplet that they're associated with in the real world. And this idea, I should just say, isn't new, but what's new is this quantification of it. So next steps here, as I've said previously, was to consider the applicability of the reference data set we're using. So in the first instance, this likely involves a better characterization of the spectral diet of human observers. We can then use the new metric we defined, the PSRM, to determine the theoretically optimal spacing and bandwidth of primaries in a multiple primary display, which will be useful for informing future display design. And in future, we might want to move towards more perceptual metrics for the non-visual light mediating mechanisms. So in the same way we have things like CIE lab space and color rendering index, we might want to define, say, a sleepiness index or alertness metric. But at present, we don't have a clear link between the photoreceptor activations and these high-level functions. Okay, so to recap what I've talked about today, firstly, I very briefly overviewed the human response to light and what aspects of that response are captured by current display technologies, and how multiple primary displays offer the ability to explicitly control the rod and melanopsin signals. I then gave a brief introduction to the RealVision MPHDR, which allows for melanopsin isolating experiments with a high dynamic range and spatiotemporal control. And finally, I touched on the limits of existing display metrics to characterize the performance of multiple primary displays and propose a novel metric that might be helpful in aiding design of multiple primary displays. And that's all I have time for today. Just want to say thank you to my collaborators on these projects, to my funders, and to the Oxford Perception Lab for being such a good group to work with every day. And finally, thank you for listening.